Welcome to the deep dive. You've sent us, well, quite a fascinating stack of sources today. We're peeling back the layers on something with a huge, often kind of unseen impact on pig health, which is, a vital part of our global food system. Our mission today is to unpack the details of one specific bacterial infection. It's called Mycoplasma hyopneumoniae, or MHyo for short, and the chronic lung disease it causes in pigs, Enzootic pneumonia. We're going to explore why this one tiny bacterium is such a big economic challenge, how it works on a molecular level, and what it means for, well, the bigger picture of animal health and even sustainability.
Yeah, it's really fascinating how something so small can have these incredibly widespread, complex effects, often setting the stage for much, much bigger problems down the line.
Yeah.
We'll be looking at its pretty unique biology, how it spreads, the ways it impacts pigs, which can be surprisingly subtle, and of course the strategies used to try and manage it.
Okay, so get ready for a deep dive into the world of swine respiratory health. Let's find out why this specific pathogen is considered kind of a linchpin in a much larger health complex. All right, let's dig in then. We're talking Mycoplasma hyopneumoniae, MHyo, the main cause of this chronic respiratory thing in pigs, in zoonotic pneumonia. Our sources really hammer home its economic importance, chronic coughing, reduced growth rates,
Yeah.
that kind of thing.
Absolutely. And this bacterium, it's really quite unique. The big thing connecting this to the wider picture is that it completely lacks a peptide-glycan cell wall. Most bacteria have one. This structural difference, it sounds technical, but it has huge implications. It makes MHyo naturally resistant to a whole class of antibiotics, things like penicillins, cephalosporins. They just don't work because they target that cell wall synthesis MHyo doesn't have.
Wow, okay, so it's intrinsically designed to dodge some of our main treatments, a sort of biological ghost in the machine. And it's pleomorphic, no fixed shape, and osmotically fragile outside a host. What exactly does osmotically fragile mean? And, how does something like that even survive?
Right. Osmotically fragile just means it's really vulnerable to changes in water pressure. Pop goes the weasel, basically, if it's not in the right environment. Which, yeah, raises the question, how does it survive? To compensate, MHyo does something pretty clever, if you can call it that, molecular theft. It actually steals cholesterol and other sterols directly from the pig's own cells, and it incorporates them into its own membrane. That makes it surprisingly tough. It really highlights that it's an obligate parasite. It has to live inside a host because it can't make these essential bits itself. Plus, it has one of the tiniest genomes known for a self-replicating organism, super specialized for this parasitic life.
Okay, so incredibly efficient in a pretty devious way. But even though it's fragile in the environment, the sources say it's highly contagious. And here's where it gets really interesting. The main transmission is respiratory aerosols, like droplets in the air. And apparently it can travel on the wind up to 3 kilometers. That's almost 2 miles.
Yeah, that airborne transmission is a huge headache for biosecurity on farms, a really significant risk. And of course, direct pig-to-pig contact, especially when they're crowded, like in nurseries or finishing barns, that helps it spread too. Another really critical point is that some pigs can be carriers without showing any signs. They just keep shedding the bacteria for a long time too, which keeps the infection cycle going, especially, like you mentioned, from sows to their piglets during lactation.
So once this sneaky little bacterium gets inside a pig, how does it actually cause the disease? What's its attack plan?
Well, the first and absolutely most critical step is adhesion, sticking on. MHyo specifically targets and latches onto the cilia, those tiny hair-like structures lining the respiratory tract. It uses special proteins like P97 and P102 to get a good grip. This stops it from just being swept out by the normal cleaning mechanisms.
Okay, it sticks. And then what? Does it just hang out there?
Oh no, it gets worse.
Okay.
Once it's attached, it starts causing direct damage to those cilia. This leads first to ciliostasis. The coordinated beating action just stops, then the cilia clumps together, and eventually they're lost completely. This basically paralyzes the mucociliary apparatus, the lung's primary mechanical defense system. It's like shutting down the escalator that clears out debris.
Right, so if that first line of defense is just gone,
Yeah.
what happens next in the lungs?
Well, that breakdown is really the central event in the disease. Mucus, dust, other inhaled stuff. It all just accumulates, and that creates this perfect environment for other pathogens to move in. In fact, MHyo is widely seen as a gateway pathogen. It damages the lungs in a way that makes pigs much more susceptible to other, often more severe respiratory infections.
That makes perfect sense. It breaks down the indical security, holds the door open for other trouble. Our sources also talk about it being a master of immune modulation.
Mm.
What does that mean for the pig's own defenses? How does that make things worse?
Yeah, immune modulation is key here. It means M. hyo doesn't provoke a strong, effective immune response that would normally clear an infection. Instead, it triggers this chronic, smoldering, localized inflammation. And paradoxically, that inflammation itself actually contributes a lot to the lung damage. You get this heavy buildup of immune cells around the airways and blood vessels, these characteristic parabronchiolar and perivascular lymphocytic cuffs. They're basically microscopic signs of this ongoing but ineffective immune battle.
So the pig's body's fighting back, but it's a response that doesn't really work and actually makes the lung damage worse over time.
Exactly. Precisely. And M. hyo can hang around in the respiratory tract for, well, up to 7 months, despite this ongoing immune response. It also seems to suppress some key immune cells while activating others. It's a very specific manipulation. This whole combination leads to the chronic nature of the disease, and a big chunk of the lung damage isn't directly from the bacteria, but from the pig's own prolonged, misdirected inflammatory reaction. It's a classic case of immunopathology, the immune system causing harm while trying to help.
Okay, given all that internal damage and sneaky immune manipulation, what does it actually look like in a pig? Are the signs obvious?
Well, in the classic form, just uncomplicated enzootic pneumonia, the most characteristic sign is a persistent cough. It's usually dry, non-productive. People often describe it as a sort of barking cough. You'll notice it more when pigs get disturbed, maybe when they move around or at feeding time.
So not necessarily a pig that looks terribly ill, maybe no fever or anything, just this chronic cough.
Yeah.
That sounds like it could be easy to miss or dismiss.
Exactly. Other signs like fever or being off-feed, lethargic, those are often absent unless, and this is key, secondary infections set in. Often the biggest impact is actually subclinical, stuff you don't easily see day to day. Things like reduced average daily gain, they just don't grow as fast. And poorer feed conversion, they eat more food for the weight they put on, which means more days to reach market weight.
Right. But the sources also mention that if it hits a herd that's never seen it before, a naive herd, it can be much worse.
Oh yes, definitely. In those herd breakdown situations, it can be quite dramatic. Pigs of all ages can get severely ill. You can see severe pneumonia, real difficulty breathing, fever, and unfortunately even high death rates. And if you look at the lungs post-mortem, the lesions are pretty typical. You see these well-defined, consolidated areas. They look firm, purplish or reddish-gray, usually concentrated in the lower front parts of the lungs, the cranioventral lobes. And microscopically, you see those lymphocytic cuffs we talked about. That's the hallmark sign.
Okay. What makes a pig more likely to get this? What are the risk factors?
Well, several things stack up. Poor ventilation in barns is a big one. Overcrowding, definitely. Any kind of environmental stress, really, things like weaning or moving pigs around during transport. And probably the most important factor is having other infections at the same time, especially viruses like PRRSV, porcine reproductive, and respiratory syndrome virus or swine influenza.
Right, and that brings us back to M. hyo being this linchpin or keystone pathogen, its role in the porcine respiratory disease complex, PRDC. What exactly is this complex?
Yeah, PRDC. It's important to understand it's not one single disease caused by one bug. It's a syndrome, a multifactorial problem involving several different pathogens, usually viruses and bacteria, acting together. And the resulting disease is typically way worse than just the sum of what each pathogen would do on its own. M. hyo is considered a really crucial primary player because, as we discussed, it damages those lung defenses. It basically creates an open invitation for all these secondary invaders.
So it sort of primes the pump, weakens the defenses, and then other bugs jump in and cause chaos.
That's a great way to put it. It primes the pump. The interaction between M. hyo and PRRSV is particularly notorious. Studies clearly show M. hyo makes the pneumonia caused by PRRSV much more severe and lasts longer. When they're both present, you see way more extensive lung damage, worse clinical signs. It's more than just additive, it's synergistic damage, driven by an over-the-top inflammatory response. And M. hyo also paves the way for bacteria like Pasteurella multicida to cause really severe, sometimes fatal, secondary bacterial pneumonia. It really does break down the door.
Okay, so given how subtle it can be initially and how complex these interactions are, how do vets actually diagnose M. hyo and enzootic pneumonia?
It really requires putting multiple pieces together. Clinical signs, like that chronic cough in a group of pigs, are suggestive, sure. And the lung lesions seen at necropsy are pretty characteristic, as we said. But M. hyo itself, it's incredibly slow and difficult to grow in the lab, so traditional bacterial culture just isn't practical for routine diagnosis.
So what's the main tool then? How do they reliably detect it?
The go-to method nowadays is PCR polymerase chain reaction. It's highly sensitive and specific for detecting M. hyopneumoniae. You can run PCR on samples like lung tissue or fluids washed from the trachea and bronchi, or even just swabs from the throat area. But, and this is a really important, but a positive PCR just tells you the DNA is there. It doesn't automatically mean the pig has active clinical disease right now. Remember, pigs can carry and shed M. hyo for months without being sick. There are also blood tests, like ELISA, that look for antibodies. These tell you if the pig has been exposed before. But the antibody response can be slow to develop, and these tests can't easily distinguish between, say, natural infection, vaccination, or antibodies passed from the mother's sow. So it's complex.
Okay, so diagnosis is tricky. What about treatment? And, maybe more crucially, prevention?
Treatment. Well, MHIO is susceptible to certain antibiotics. Things like tylosin, tiamulin are commonly used. They can be effective, especially if you treat early, in reducing the clinical signs, like the coughing. But antibiotics usually don't eliminate the organism completely. The pig often remains colonized, a potential carrier. And of course, supportive care is important too. Good air quality, not overcrowding them. That helps manage the impact.
Right. So if treatment isn't a silver bullet, prevention must be absolutely paramount. What are the main strategies there?
Vaccination is definitely the cornerstone of control in most commercial herds. There are several good vaccines available. They work primarily by reducing the severity of clinical signs, cutting down on the extent of lung lesions, and ultimately improving growth performance. But it's really important to understand what vaccines don't do. They generally don't prevent pigs from getting colonized by M. hyo in the first place. And they don't completely stop vaccinated pigs from shedding the bacteria either. The goal is more about controlling the impact of the disease, mitigating the economic losses.
Okay, so the vaccines help the pig and the herd cope better rather than making them completely bulletproof to the bug itself.
Exactly. That's a good way to think about it. And beyond vaccination, strict biosecurity is vital, non-negotiable, really. Things like all-in, all-out management, where you completely empty a barn, clean it, disinfect it before a new group of pigs comes in. That helps break the infection cycle. Also, carefully isolating and testing any new animals coming onto the farm. Improving ventilation is huge, reducing dust, ammonia levels, and actively managing those other co-infections we talked about, like PRRSV. There's also a lot of focus on stabilizing the breeding herd, the sows. Things like gilt acclimation programs, making sure replacement females get exposed in a controlled way before they enter the main herd, and so vaccination protocols. The idea is to build up a more uniform level of immunity in the sows, which helps reduce shedding and transmission to their piglets.
Wow, it really is a multi-layered continuous effort then, not just a one-shot fix.
Yeah.
Okay, we've gone through the subtle signs, the complex damage it does, but let's talk bottom line. What's the real-world impact of MHU on the pig industry?
Oh, the economic impact is huge, truly profound. M. hyo infection, or Enzootic pneumonia, is consistently ranked as one of the most economically damaging diseases in the global swine industry. Just in the U.S., estimates put the annual losses somewhere between, say, $375 to $400 million, which makes it second only to PRRSV in terms of sheer economic cost.
That is. Yeah, that's a staggering number. Where do those losses actually come from? How does it add up?
The biggest single drain is from reduced growth performance. That's the main driver. Pigs with enzootic pneumonia just don't grow as efficiently. One study calculated they lose about 37 grams of average daily gain for every 10% of their lung tissue that shows lesions. They also have worse feed conversion ratios, meaning they eat more feed for every pound or kilo of weight they gain. And that means they take longer to reach market weight, sometimes an extra week or so on feed, which costs money and often results in lighter carcasses at slaughter.
So it's slower growth, wasted feed.
Yeah.
That adds up incredibly quickly across millions of pigs.
It really does. And then you have the direct costs of medications and vaccines. You have increased mortality rates, especially when those secondary infections like Pastorella get involved. And sometimes even parts of carcasses or whole carcasses getting condemned at the slaughter plant due to severe pneumonia lesions. And what's really telling, connecting back to PRDC, is the synergistic cost of co-infections. For example, the cost impact of having both PRRSV and M. hyo together has been estimated at nearly $10 per pig. And that's about 1.5 times more than just adding up the individual costs of PRSV alone and M. hyo alone. It's a multiplier effect.
That's a really powerful illustration of how M. hyo acts as that keystone or gateway we keep mentioning. Are there other impacts, maybe less obvious ones?
Well, this raises a really important point, actually, for you listening and for the wider world. Beyond the direct dollars and cents, people have started looking at the environmental footprint, and M. hyo has one. Because of that reduced feed efficiency and the extra days needed to raise the pigs, it means more resources are consumed per unit of pork produced. Some estimates suggest M. hyo infection increases the carbon footprint of pork production by maybe 2% to 4%, and water usage by maybe 2.5% to 4.1%. So it really highlights that controlling diseases like this isn't just about economics or animal welfare, though those are critical. It's also a really key part of making pork production more sustainable.
So wrapping this up, what have we learned? We've taken quite the deep dive into Mycoplasma hyopneumoniae and Enzootic pneumonia. We've uncovered its really unusual biology, no cell wall, its sneaky tactics for damaging pig lungs, and its critical role as that gateway pathogen in the bigger porcine respiratory disease complex. We've seen how its subtle early signs can hide this massive economic drain, driving up costs, reducing efficiency, and even contributing to a larger environmental footprint.
Yeah, I think this journey really shows just how complex these interactions between a pathogen and its host can be. And understanding all the details, from the molecular theft of cholesterol right through to the immunopathology, the way the immune system itself causes damage. That understanding is absolutely vital if we want to develop truly effective control strategies. It also just hammers home how interconnected everything is. Animal health, the economics of agriculture, even these big global challenges like sustainability.
Right. So here's a final thought for you to chew on. If a bacterium as seemingly simple as M. hyo that doesn't even have a proper cell wall can cause such complex, widespread, and economically devastating problems for an entire global industry, what other silent, perhaps even simpler seeming threats might be lurking out there in the complex biological systems we depend on, just waiting for the right conditions to emerge and reveal their true compounding cost?